Welcome to Brainstorm, where we give you a glimpse into the world of science for this Monday, June 18th, 2012. First, we turn to the world of medicine. A study led by Swedish researchers has shown positive effects for an Alzheimer's vaccine. Before we get into the vaccine, let's first go over the actual cause of Alzheimer's and the resulting dementia associated with it. Now, the exact mechanism isn't fully known, but current knowledge points to amyloid precursor proteins. In people with Alzheimer's, these proteins build up and form a substance called beta amyloid. As this substance builds up, it forms plaque around brain cells, eventually leading to cell death. Currently, the only medications that target the disease attempt to alleviate symptoms, but don't actually treat the cause. Almost a decade ago, a clinical trial was done on a different potential vaccine. Unfortunately, that old vaccine caused the immune system to attack brain cells. The trial was obviously discontinued. But this most recent trial has lasted 3 years, the newer vaccine resulting in no noticeable side effects. The success is from the more specific targeting of the beta amyloid substance, with 80% of test subjects producing active antibodies against it. Hopefully, this could become a treatment for mild to moderate Alzheimer's, although larger trials are needed. Our next story is from the world of neuroscience. Scientists at the Gladstone Institute recently turned skin cells into neural cells. Now, usually when we discuss scientists converting one cell type into another, it's through something like induced pluripotent stem cells, those being adult cells altered to act like embryonic cells, able to differentiate into any cell type. This, on the other hand, was a direct conversion, turning skin cells into neural stem cells without going through a pluripotent stage. Previous work had been able to accomplish this transformation using a combination of compounds and genetic factors, but this latest work used just one. Not only did this turn skin cells into neural stem cells, after a few multiplications, the resulting neurons formed a primitive network within a petri dish. To test the safety of these cells, the grown mouse neurons were transplanted back into the mice. Like the scientists suspected, skipping the pluripotent stage made the new cells safer to use. And while it'll be a while before humans are regrowing bits of brain, this development has more immediate applications. Specifically testing drugs, as many current models don't accurately predict what a drug will actually do in the brain. Further research will go into generating specific neuron subtypes, allowing for detailed testing of a drug's safety and effectiveness. We end with a quick update from the world of biotechnology. A group at UC Irving are developing a potentially valuable tool in the fight against malaria. As we discussed a few weeks ago, malaria is a serious threat, with 40% of the population in regions where it's a risk. That story was about a potential vaccine made by algae, but what if we could directly limit the spread of malaria? To achieve that, scientists have actually made transgenic mosquitoes that impair the development of the malaria parasite. This means the mosquitoes actually can't spread the disease because the parasite gets too weakened by the modified mosquito's immune response. The exciting thing is that the modifications don't appear to affect the insect's fitness, meaning they could potentially be introduced into wild populations around the world. Now, this immune response was actually engineered to mimic that of mice exposed to human-targeted malaria, as they actively produce antibodies that kill the parasite. Hopefully, this research will be expanded upon and implemented quickly to reduce the spread of this deadly disease. Well, hope you enjoyed this episode. If you did, please consider subscribing and be sure to check the links in the video description. The first link is about a new series on this channel starting this summer. There's less than a week left to raise funds, so definitely check it out.